Any questions?
Yeah, so what's the underlying source of these studies? Is it basically straight from PubMed or?
For this stuff? Yeah. Yes. So this would be more, that's why, I mean, the canonical stuff, the pathways are based on mostly the literature for sure. Yep. So this is the part, and that's the pathway enrichment. It's more of a text mining. The other stuff, you're kind of correlating. You're finding out, I have all these bits of data associated with a study. How is it similar to this other bit, right? Here we're just saying, I have all these genes. What pieces of text is overrepresented associated with all those genes?
And Mike, just one more comment on the source of the data. One thing that's a little bit different here is the actual data itself is what's being used to generate what we're looking at. And it goes through a curation process. So this Illumina acquired a company that spent a lot of time and effort curating data, and the majority of the studies they reviewed actually don't meet the quality thresholds. That might be a surprise to some who read a lot of primary research, but no, these were curated by scientists in the Next Bio platform. They continue to do that now. I get emails every week about the new studies that they add to the platform. So they're still doing it. And there's close to 10 people, and they've been working on it for almost a decade, and they're just curation scientists. So that's really the source. They're searching public databases and manually curating it and putting the data itself into the system.
That's kind of where the future of this stuff is going. Like, it's all, I mean, it just doesn't pay to just, having an N of 10, when everybody's going to have their DNA sequence at some point, we're going to have access to thousands and thousands of different samples. It just doesn't pay to just look at one little study, right? I mean, this is where bioinformatics is going. I think people use me in the beginning to basically say, hey, I have a problem. I want you to help me solve it, on this problem. But I think what's going to happen, it's going to flip, is that all this information is out there. You're going to have people mining the data going, hey, I looked at breast cancer and I think this gene is important. Where are my experts that work on that? Like I said, this can save you years at the bench. And I started before, this stuff really came online, and it was hard. It was a lot of stabbing in the dark, running just, Westerns or, running, thousands upon thousands of RT-PCR, the nice thing is you don't have to spend a dime and you can already get there with this stuff. But I also want to stress too is that, you should not let the computer make your decisions for you. And, I always say this talk or say this story when I give this talk or this example is that, everybody knows that Big Blue, the IBM supercomputer, can beat the world's greatest chess player at chess, right? Everybody knows that. Yeah, and when you first hear that, you're like, oh, computer wins, yay. But what they don't tell you is that that person can get a computer, that chess player can get a computer and beat Big Blue, right? It's not either the computer or the human's better. It's when you combine both, it's better than both. That this isn't a pipeline. At this point, what I am telling you now, some of the processing stuff is total pipeline. Which is probably why I'm not very much interested in it. It's this that I'm interested in because this is the art form. There is no follow A to B to C to D. It's here's all the data. Your system. Play around. Find something. And when you find something, pull it out and test it. If it don't work, find something else. What you can also do is once you find something and then you prove it, now you've got another anchor and now you can go somewhere else. And I do this stuff all the time is that I swear half the stuff I analyze, it isn't even the people that I collaborate with. It's not their research. It's somebody else's. And a lot of times what I find is when I'll work on somebody's data set and I'll find that it's not very good, which happens quite a bit. And so I'll say, hey, instead of scrapping your data, scrapping your project, why don't we go out in there and find some other data? And sure enough, it'll be better than theirs, way better. And I'll look at them, at first they, very hesitant. Scientists are very, this is all mine, don't scoop me, wants to hide. But, once they start getting into this, they realize once you start looking at other people's data, you realize the true power is not to analyze your stuff, it's to analyze everybody else's stuff, because there's way more data than you could ever potentially generate just from yourself. Any questions? Yeah.
So when you generate a gene list, it doesn't really make sense. So for example, you're looking at heart disease, and what you see are genes that are for, say, cancer. How do you go about brainstorming to try to understand what's actually going on?
Here's what I would do, is that if you had, say you were looking at heart disease and you caught up with a bunch of stuff with cancer, and that happens a lot, right? Anytime you're remodeling tissue, a lot of cancer stuff's going to come up. And a lot of these databases, they're filled by us, right? What do a lot of us researchers do? Cancer research. So there's a lot of cancer stuff, right? It comes up. Here's what you would do is I would take your heart study and then I would take a cancer study, do a Venn diagram, find out what's common, then just take the ones that were common with the cancer stuff, and then everything else left behind in your heart stuff should be should be good. Questions? Yeah.
I noticed in your analysis, CA-153 is a marker which we use clinically all the time for testing breast cancer. But that didn't show up.
Which one?
CA-153. My question is, does this always match with current diagnostics markers we use?
I would say a lot of times it does, but sometimes it doesn't. And I would say when it doesn't, that's actually a little more exciting, in my opinion. Yeah.
Because we can prove that some of the markers we use are probably not the best markers.
Totally. And that happens to me all the time. Or here's what happen is I'll find a marker like this marker here, the one we were looking at, the, what was that one? It wasn't arachnid. It was the one that first came up at the top. I'll find, I'll work on breast cancer, say, and I'll find a gene and it'll say, oh, this is the greatest marker for colorectal cancer, right? And there'll be nothing to have that says anything about breast cancer, right? A lot of times what I'll do is I'll find a great marker, but it won't be known in that field, right? And that does happen for sure. It is, and the nice thing about this stuff, it's all going to get more powerful the more data that gets deposited into it, right? That is a nice thing, is that, that the 20,000, the thing you saw on the next or the Illumina or the correlation engine, most of that stuff is curated from PubMed. How many studies did I say that PubMed had? Or not PubMed, Geo. Does anybody remember the list of how many studies were in Geo? About 70,000, right? They've pretty much gone through that at Illumina. They're now down to 20,000. About 75% of everything in the Geo isn't passing the quality control, right? And it's not only that, it's papers that, with grants and people trying to get grants and funding so lax, there's a lot of garbage out there. There really is. And what I like about these kind of tools is I don't have to believe what they say, right? I can go get their paper, and I can read it and say, oh, okay, well, I'm going to see if you're right, and I'm going to go get their data and use this tool and kind of analyze it and see if what they say is true. Any other questions? I'll leave you with, I work on my own. I'm actually going to be hanging out for a little bit after this. If you want to talk about your bioinformatics project or any of your projects or some of the tools available, I'll be around. So just come find me. Also, as I say, I do consulting. So if you see this and you go, hey, it's cool, but I don't want to have to I don't want to learn any of this stuff, you can always get me and I can do it for you. Although I'm not free. All right. Well, thank you very much. Appreciate it.